So continuation with the yesterday's class, where we have seen the uh, three important pathways from the carbohydrate metabolism from the presentations, that is the glycolysis, gluconeogenesis, and the PCA cycle. And now the part 2 of this module uh, is nothing but the electron transport chain and oxidative phosphorylation. So in order to understand the electron transport chain and oxidative phosphorylation, so we need to understand the bit of the basics of the bioenergetics. So we know bioenergetics, it is a branch of thermodynamics, mainly deals about the change in the energy and how the energy is transformed from one form to another form. But it doesn't say that how fast the rate of reaction. It can only say that thermodynamics can only say that whether the reaction is spontaneous or non-spontaneous, but it does not say anything about the rate of the process. And in thermodynamics, we have three systems. One is the isolated system. Second one is the closed system. Third one is the open system. So here system means the object that is being studied, whereas surrounding means other than system, everything else in the universe. So in isolated system, there is no exchange of matter or heat between the system and the surroundings. You can see here that in isolated system, no exchange of matter or the heat between the system and the surrounding. But whereas in a closed system, there is the exchange of heat, but not the matter between the system and the surrounding. So that means heat exchange takes place, but not the matter. And coming to the open system, in open system, heat as well as matter exchange takes place between system and the surroundings. All living organisms or the living cells are open systems because the exchange of both material and energy with the surroundings takes place. So now coming to the, we know that the cell needs energy for their various activities for assembling the smaller molecules into the polymeric molecules. And it also requires energy for all the functions like living, growing, reproducing, and so on. So therefore, it is useful to describe the energy in biological systems using the thermodynamic terminology of thermodynamics. So we know that the according to the first law of thermodynamics, the energy is highly conserved, so it cannot be created or it cannot destroy. It can only transform from one form to another form, and that is the first law of thermodynamics. And second law of thermodynamics is that the universe tends to towards the maximum disorder. So in the sense, it deals about the randomness or the disorderedness of uh, 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 the disorderedness of this one. Then we know we since to understand the electron transport chain, we also need to understand the terminology we come across in thermodynamics, that is the free energy, enthalpy, and entropy. So we know that free energy, that is energy that is available to do work. So this, we can also call it the energy that is relevant to biochemical systems, and this free energy is abbreviated with G, and the units is joules per mole. And the free energy has two components. One is enthalpy, another one is entropy. The enthalpy is the heat content of the system, whereas the entropy is the randomness or the disorderedness of the system. So, so the in the relationship between the enthalpy and entropy and the free energy is that the G is equal to H minus T, yes, H minus T, yes. So that means G is equal to H minus Ts, and here H is the heat content, that is enthalpy, S is the desiredness, and so therefore delta G is equal to delta H minus T delta S. And if the delta G is less than 1, what happens if the delta G is greater than, sorry, if the delta G is less than 1, less than 0, what happens if the delta G is greater than 0, what happens if the delta G is 0, what happens if the delta G free energy change is greater than 0, then that reaction is non-spontaneous. If the delta G is less than 0, then it is a spontaneous reaction, and if it is 0, it is equilibrium. So I think this you have already studied. And so with this background, we try to understand the electron transport chain. And in the electron transport chain, we need to understand how the high energy molecules are the ATP, and ATP is synthesized from reducing equivalents like NADH and FADH2 that are produced in the cell. So we know that in all aerobic organisms, the oxygen is utilized for the aerobic respiration. So that means for the complete oxidation of one glucose molecule, for example, you can see here C6H12O6 plus 6O2 gives us a 6CO2 plus 6H2O. This is the complete oxidation of the glucose. So this reaction has two half reactions. First, when the carbon atoms in the glucose are oxidized and releases the electrons, and these electrons are further reduces the molecular oxygen to water. So that's what happens. But only thing is, when the glucose is oxidized, when the carbon atoms in the glucose are oxidized, the electrons are released. These electrons are trapped. These electrons are trapped and in the form of reducing equivalents like NADH and FADH2 and so on. These NADH, FADH2, when they again release, transfer their electrons to the molecular oxygen through several oxidation reduction center in the electron transport chain, the high energy molecule ATP is produced and the oxygen becomes water. And so this is the process that takes place in transport electron transport chain. Now coming to this one, we know that in electron in when the glucose undergoes oxidation in the glycolysis to pyruvate, and this pyruvate is further converted into the acetyl-CoA by pyruvate dehydrogenase complex. This acetyl-CoA further enters into the TCA cycle, and then so in the TCA cycle again it produces the NADH and FADH to reducing equivalents. So therefore, from one molecule of glucose, from the one molecule of glucose, the the NADH and FADH2 molecules are produced both in the glycolysis and the TCA cycle, and all these NADH, 10 NADH and 2 FADH2 molecules, all these are high energy molecules. So they have the high energy electrons in their structure, and these high energy electrons are transferred to the molecular oxygen during the electron transport chain for the synthesis of high energy molecule ATP. So that means this is the process that takes place, because all these 10 NADH and 2 FADH2 molecules are the high energy reducing equivalents, and they transfer their electrons to the molecular oxygen through electron transport chain, and in order to form the high energy molecule ATP. So now, so that means that means in the electron transport chain, three events takes place. One is the high energy molecules that are produced in the glycolysis and TCA cycle, like NADH and FADH2. These molecules should undergo oxidation. They should to NAD plus and FAD, so that they can participate in the additional substrate oxidation reactions. Oxidation reactions. Second one is when NADH and FADH2, when they are oxidized, and these electrons are transferred to the molecular oxygen through various oxidation reduction centers that are present in the inner mitochondrial membrane. And this you can also see in this diagram. You can see here that this is the NADH, and here the FADH is formed here. That is the when succinate is converted into the fumarate, and this FADH to NADH, when they are transferring their electrons to the molecular oxygen, you can see here. So there are many oxidation reduction centers are involved in between, and during many these all these complexes have the many oxidation reduction centers. So during this process, then the electrons are transferred from NADH and FADH2 to the molecular oxygen through various oxidation reduction centers that are present in the mitochondrial membrane. And during this process, the protons are expelled from matrix to the outside. You can also see here the protons are expelled. As a result, the proton gradient is formed. You can also clearly see here that during this electron transfer, the protons are expelled. As a result, the proton gradient is formed, and this proton gradient is very essential for for synthesizing ATP, because when these protons again move back to the matrix through another complex that is called ATP synthase, the ADP would be converted into the ATP. So these are the three important events that takes place in the electron transport chain. So as I already said, that the first one is the oxidation of NADH and FADH2 that are produced in the glycolysis and TCA cycle. And during this oxidation, the electrons are released, and uh, these electrons are again transferred to the molecular oxygen in the electron transport chain through various oxidation reduction centers. And during this process, the protons are transported across the membrane into the intermembrane space. As a result, the proton gradient is generated, and this proton gradient is utilized to drive the synthesis of ATP from ADP and PI by the oxidative phosphorylation. So these are the three events that takes place in the electron transport chain. And now coming to the since this process takes place in the mitochondria, and we need to see the details. And so we know that before going to explain the in details, we will just again summarize what exactly the takes place in the electron transport during the electron transport chain. As I already said, that the role of the electron transport chain is only to synthesize a high energy molecule ATP from the NADH and FADH2. These are the high energy reducing equivalents, and So this entire process takes place in the inner mitochondrial membrane, and so that means there are many enzymes and coenzymes are involved in this process. And during the transfer of electrons from the NADH and FADH to the molecular oxygen, the pH gradient is formed, and this pH gradient is further utilized for the synthesis of ATP. So this electron transport chain in eukaryotes, it takes place in the inner mitochondrial membrane, whereas in case of prokaryotes, it takes place in the plasma membrane. And now, since there are many complexes are present in the inner membrane to carry out this reaction. So during the electron transport, so as I already said that when NADH is donating its electron to the terminal electron acceptor, that is a molecular oxygen, the oxygen is reduced to water, and the proton gradient would be generated, and this proton gradient in turn drives the ATP synthesis by ATP synthase, and this is also known as the oxidative phosphorylation. So that means the during this process, during the during this process of electron transport chain, once again you remember that the proton gradient is generated. So this proton gradient, in the sense, this is the matrix, this is the intermembrane space, and when the electrons are transferred from NADH and FADH to the molecular oxygen through oxidation reduction centers that are present in various complexes in the inner membrane, the protons are translocated across the membrane, and these protons are essential for the synthesis of ATP from ADP, because by ATP synthase. So we will see the details one by one. So now, since this process takes place in the mitochondria, and we need to go through the structure of the mitochondria. So we know that mitochondria is the storehouse of the cell, and many aerobic reactions that takes place, mainly the citric acid TCA cycle and fatty acid oxidation, and also in both the cases the FADH and the reduced equivalents are formed, and these reduced equivalents are further undergoes oxidation uh, in the electron transport chain by various complexes. And since the mitochondria is the storehouse of the cell, and we know that the number of mitochondria varies from cell to cell, and it, of course, it depends on the energy requirement of the cell. For example, unicellular algae has only one mitochondria, whereas the mammalian liver cell has the 5000. And if you see, the white muscle contains very few mitochondria, whereas the red muscle tissue has many mitochondria. And so that is the reason why the alligator jaw muscle is the example for the white muscle. So the alligator can snap their jaws with astonishing speed and force, but it cannot continue this motion beyond a few repetitions. In the sense, because it has less number of mitochondria in the white muscle, whereas the migratory birds, they have the red muscle, and the migratory birds can able to fly many thousand kilometers because the red muscle has many mitochondria, and that's why the migratory birds also require high energy for their locomotor motion. That's why the migratory bird, the red muscle present in the migratory birds, have the higher mitochondria. And so if you see its structure, the size of the mitochondria is 0.2 to 0.8 micrometer, and the length is 0.5 to 1.5 micrometer. It has two membranes, outer membrane and the inner membrane. Outer membrane is it has the porins, and it allows the water soluble molecules very freely, whereas the inner membrane is highly impermeable to the charged and the large polar molecules, and it is only permeable to the uncharged molecules such as water, oxygen, and carbon dioxide. So that means only outer membrane is permeable, whereas the inner membrane is impermeable to the large polar molecules as well as the charged species. And so in addition to this, the it has so inner membrane has many transport proteins, and these transport proteins are involved in transporting many polar charged molecules as well. So the inner membrane is highly folded, that is known as the cristae, and the matrix contains many enzymes, that is pyridihydrogenase complex, citric acid cycle enzymes, enzymes of fatty acid oxidation, and so on. And so now, so coming to since the electron transport chain takes place in the inner mitochondrial membrane, and this inner mitochondrial membrane has many complexes for facilitating the electron transport chain. There are 4 oligomeric complexes are present in the inner mitochondrial membrane. They are named they are also called as complex 1, complex 2, 3, and 4. In addition to this, there is another complex, that is complex 5, that is also known as the ATP synthase. All these complexes you can see here, 1, 2, 3, 4, and this is the ATP synthase is complex 5. All these complexes contain a variety of oxidation reduction centers, and these oxidation reduction centers may include the cofactors such as FAD, FMN, and ubiquinone. In addition to this, other centers like iron sulfur clusters, cytochromes, and copper proteins are also present. And in electron transport chain, the electron flow takes place via the oxidation, various oxidation reduction centers present, that is from NADH2 to the molecular oxygen, and it always takes place from the reducing agent to the oxidizing agent. And now, so coming to this one, in electron transport chain, all these complexes, complex 1, 2, 3, 4, 5, all the 4, all these complexes are arranged in such a way that they are arranged in such a way that they start from the negative redox potential to the positive redox potential. So in the sense, the more the negative, the complex can easily lose the electron. The more the positive, the complex can easily gain the electrons. So such arrangement facilitates the unidirectional electron flow. So that means that all the electron transport chain complexes are arranged in such a way that they start from negative redox potential to the positive redox potential. And in addition to these complexes, there are some other coenzymes present. These are ubiquinone and the cytochrome C. These are also known as the mobile electron carriers. You can see here, this is the ubiquinone and this is cytochrome C, and they are not the part of the complexes. They are the mobile electron carriers. They can take the electrons from one complex and transfers to the other complex. So these two are the mobile electron carriers in the electron transport chain. So the ubiquinone is it is a lipid soluble molecule, and this ubiquinone transfers the electrons from complex 1 to 1 or 2 to complex 3. You can see here in this diagram, this is ubiquinone. It can take electrons from the complex, either complex 1 or from complex 2, and it can pass it on to the complex 3. So that's why it is called as a mobile electron carrier. In the same way, cytochrome C, it can transfer electrons from the complex 3 to complex 4. And among ubiquinone and cytochrome C, ubiquinone is a lipid molecule, whereas the cytochrome C is a protein. That is only the difference between the ubiquinone and the cytochrome C. And so when the electrons are transferred from the NADH, to the molecular oxygen, the energy is released, and that energy is utilized for the generation of the proton motive force across the membrane, and this proton motive force is further utilized for the formation of ATP from ADP and PI. So this is the overview of the this process. And of course, you can also see here that the complex 1, complex 3, and complex 4 are involved in the translocation of protons, because they have the proton translocating activity, whereas the complex 2 is not involved directly in the proton translocation. In the sense, during electron transport chain, the energy liberated at the complex 2 is not sufficient for the for expelling the protons from the matrix to the intermembrane space. So that therefore the complex 2 is not directly involved in the proton translocation. And you can let me make it clear that the complex 1, when the electrons are transferred from NADH to the complex 1 and complex 1 to complex 2, the complex 1 is involved in transporting 4 protons from matrix to the intermembrane space, whereas the complex 3 is involved in the translocation of the 4 protons from matrix to the intermembrane space, whereas complex 4 complex 4 translocates the 2 protons. So in the sense, the exact mechanism of how the translocation is not very clear, but the complex 1 can translocate 4 protons, complex 3 causes the translocation of 4 protons, and the complex 4 causes the translocation of 2 protons. In the sense, these translocation of these protons are only when one pair of electrons are transferred from NADH to the molecular oxygen. This we will make it clear in the later. And so therefore, so that means during this translocation of when the electrons are transferred to the NADH2 and FADH, NADH and FADH2 to the molecular oxygen, the proton gradient is generated, and that proton gradient is in turn used to the synthesis of the ATP by the ATP synthase. And these are the 4 complexes, as I already said, that complex 1 NADHQ reductase, complex 2 succinate Q reductase, complex 3 cytochrome C reductase, and the complex 4 cytochrome C oxidase. And these are all cytochrome C, it is a mobile electron carrier. And you can also see here that all these complexes have several cofactors. So for example, coenzymes, you can see here complex 1 has FMN as a cofactor, whereas complex 2 has FAD as a cofactor, whereas cytochrome C reductase has many heme proteins, heme BL, heme BH, heme C1. And in addition to this, the complex 1 and complex 2 and complex 3 also has the several iron sulfur clusters, and the cytochrome C oxidase has heme A, heme A3, copper A, and copper B. Copper B cofactors. And as I already said, that in electron transport chain, all the 4 complexes are present in such a way that they start from the negative redox potential to the positive redox potential. You can see here complex 1 is more negative compared to complex 2 and complex 3. Complex 3 is more negative compared to the complex 4. So that means, so that means the more the negative, they can easily lose the electron. The more the positive, they can easily accept the electrons during the electron transport chain. So that facilitates the unidirectional transfer of electrons in the electron transport chain from NADH and FADH2 to the molecular oxygen, and this molecular oxygen undergoes reduction to the water. So this is the process that takes place. Now we will see one by one. You can also see here all these complexes are the complex 1 is more negative, whereas the complex 4 is more positive. That's why this drives the unidirectional electron flow. And now coming to as I said, that there are many cofactors are also involved in this process. These are all mainly the FMN, FAD, and the iron sulfur clusters, and also the ubiquinone and cytochrome C. All these are the various cofactors that are involved in the electron transport chain. As I already said, that ubiquinone and cytochrome C, these are the mobile electron carriers. Ubiquinone is a lipid soluble, whereas cytochrome C is a peripheral protein, as I said in the previous slide. And so these iron sulfur cluster, this is the structure of the iron sulfur clusters. You can see here, if you see the iron sulfur clusters, and in this, there are 2 types of iron sulfur clusters present. One is Fe2S2, that is the dimeric, and second one is the Fe4S4, these are the tetrameric. And in both the cases, you can carefully observe that the iron atom is linked to the 4 sulfur atoms. 2 are inorganic molecular sulfur, and 2 are the sulfur side chains of the cysteine. And in both the cases, during electron transfer, they can take one electron, and they can also release one electron at a time during the electron transport process, because during this process the iron switches from one oxidation state to the another oxidation state. You can also see here in the Fe+3 state, when it takes one electron, it becomes Fe2 plus. Again, if it releases to the next carrier, again it undergoes oxidation back to the Fe3 plus. And so now, so in addition to this, there are some cytochromes are also present, and these cytochromes are also present. Cytochrome cofactors are also present. This we will see later. So as I already said, that ubiquinone it is also known as the coenzyme Q. Throughout the presentation, when you come across the coenzyme Q or CoQ, you should understand that ubiquinone and coenzyme Q both are same. So this ubiquinone is the mobile electron carrier. So this ubiquinone carries, it takes 2 electrons, but it takes only 1 electron at a time. So this is the ubiquinone, that is the oxidized form of ubiquinone. When it accepts 1 electron, it becomes semiquinone, and the semiquinone again accepts 2nd electron and the 2 protons, it becomes ubiquinol. So that means it accepts 2 electrons, but 1 at a time. And so now this is about the work of the electron transport chain. Now coming to the details of the each and every step that takes place in the electron transport chain, we will try to elaborate. And here first we will discuss about the complex 1. So we know the complex 1 is the NADHQ oxidoreductase. So this is the complex 1, and this is the complex 1 NADHQ oxidoreductase. And this complex 1 has, you can see here, it has many subunits, and it has FMN as the cofactor, and it has many iron sulfur clusters. You can see here. So the FMN is the cofactor, and it has 8 to 9 iron sulfur clusters. And this complex 1 it is a very complicated enzyme, and it has the structure has the proton transporter activity. As I already said, that complex 1 is involved in translocating the 4 protons. So if you see here that as I already said, that if you see the detailed the previous diagram where I showed this one, the complex 1, first what happens at the complex 1 is that initially the high energy molecule NADH, so the high energy reducing equivalent NADH donates its electron to the complex 1, and here during this process 2 electrons are donated to the 2 electrons are donated in the form of the hydride ion. And you can see here the first hydride ion is transferred to the hydride ion is transferred to the FMN, and FMN becomes FMNH2, and the FMNH2 further transfer electrons to the iron sulfur clusters, and these iron sulfur clusters further transfers to the ubiquinone, and ubiquinone becomes ubiquinol. So that means what exactly happens here is that it has many subunits. As I already said, that in prokaryotes it has 14 subunits. In eukaryotes it has 14 plus many additional subunits. And but all this it has many one FMN. The FMN here is the cofactor, and it has many iron sulfur clusters. And what happens here is that when NADH and donates its electrons in the form of hydride, initially the FMN is reduced to FMNH2. This FMNH2 again releases these electrons to the iron sulfur clusters, and these iron sulfur clusters, there are many iron sulfur clusters, and these iron sulfur clusters, in these iron sulfur clusters, the iron is reduced from Fe3 plus state to Fe2 plus state, and these iron sulfur clusters further transfers this electron to the coenzyme Q. As a result, the coenzyme Q or the ubiquinone both are same. So the ubiquinone becomes ubiquinol. So that means the electrons that are transferred from NADH, NADH is reduced the ubiquinone to ubiquinol. And this is the simplest way. You can also see here the what process takes place. The NADH, when it is oxidized, the hydrogen ion, the two electrons are transferred in the form of hydrogen ion. So here FMN is reduced to FMNH2, and this FMNH2 again releases transfer these electrons to the coenzyme Q through iron sulfur clusters. During this process, the 4 protons are translocated from matrix to the intermembrane space, and as I already said that the mechanism is not very clear that the about this translocation. And as you can see here, that as I already said that ubiquinone can accept only the 1 electron at a time, but you can see here when it accepts 1 electron, it becomes semiquinone. When it accepts second electron and the proton, it becomes the ubiquinol, or the reduced ubiquinone. And so the net reaction here is that NADH is donating its electrons, and NADH is undergoing oxidation to NAD plus, whereas the ubiquinone is undergoing reduction to ubiquinol. This is the net reaction that takes place at the complex 1. So during this process, as I already said that the four protons are translocated across the membrane, but how this proton transport occurs is not clearly known. So this is the that's what takes place in case of complex 1. Coming to the complex 2. Complex 2 is also known as the succinate dehydrogenase, or the succinate ubiquinone reductase, and it is the complex 2 is the part of the TCA cycle enzyme. So as you can see here, it is a part of the TCA cycle enzyme, and where the succinate is converted to the fumarate by the succinate dehydrogenase, and that generates the FADH2. And so therefore this the succinate dehydrogenase has contains 3 identical multi-subunit enzymes, and it has a mushroom shape, and it has the FAD as the cofactor. You can also see here, this is the FAD as a cofactor. It also contains many iron sulfur clusters, and also the heme B proteins. And here, as I already said, that first the FADH2. So FADH2 transfer these electrons to the FADH2 transfer these electrons to the FADH2 transfer these electrons to the iron sulfur clusters. You can clearly see here iron sulfur clusters. So this same diagram you can also see here. So iron sulfur clusters. These iron sulfur clusters further in turn causes the reduction of ubiquinone to ubiquinol. So in that sense, so since it is the complex 2 is a part of the citric acid cycle enzyme, and when the succinate is getting converted to fumarate and FAD is reduced to FADH2, this FADH2 directly feeds the electrons to the iron sulfur clusters. These iron sulfur clusters undergoes reduction, and then this reduced iron sulfur cluster in turn transfer electrons to the ubiquinone. As a result, ubiquinone becomes ubiquinol. So the net reaction in complex 2 is that when succinate is converted to fumarate, FAD is converted to FADH2. This FADH2, when it undergoes again oxidation back to FAD, and the ubiquinone becomes ubiquinol. So this is about the complex 2. Now coming to the complex 3. Complex 3 is also known as the cytochrome oxidoreductase, or cytochrome BC complex, and this enzyme catalyzes the oxidation of the ubiquinol and reduction of the cytochrome C. You can also see here that so this is the complex 3, and now the complex 3, what it does is it takes the electrons from the ubiquinol and it releases to the cytochrome C.
So the transfer of electrons from the ubiquinol to the cytochrome C through a complex 3 takes place by a special process known as the Q cycle. And this complex 3 has again it has three major subunits: cytochrome C, cytochrome B, and the iron sulfur protein. And you can also see here that the detailed mechanism. The transfer of electrons from ubiquinone to the cytochrome C takes place by a special process called Q cycle. So at the beginning, you can also see step by step here. At the beginning of the Q cycle, the ubiquinol so this is ubiquinol. So that means reduced ubiquinol by complex 1 and complex 2. So this ubiquinol binds to the ubiquinone binding site that is present at the intramembrane space. That is, you can also see here this complex 3 has the ubiquinone binding sites. One is at the outside, that is intramembrane side. Another one is at the matrix side. So initially, at the first, the ubiquinol diffuses into the QP, or it is also called as a QO site. And so when it binds to the QP site, you can see here. And initially, this ubiquinol transfers one electron to the iron sulfur protein. You can see here this ubiquinol transfer one electron to the iron sulfur protein, and the two protons are pumped out, and as a result, this ubiquinol becomes the semiquinone, and the semiquinone again transfers another electron to the cytochrome Bl. As a result, it becomes again ubiquinone. So that means ubiquinol after one electron it becomes a semiquinone. After a second electron release, it becomes again ubiquinone, and this ubiquinone again diffuses through the membrane. Again, it binds at the QI site. So QI site. Now again, this ubiquinone, again the electron that is released to the cytochrome Bl by the semiquinone, again further it transfers this electron to the cytochrome BH, and cytochrome BH further transfer these electrons to the ubiquinone. As a result, the ubiquinone becomes again semiquinone. So that means in the first half, this Q cycle takes place in the two half cycles. In the first half cycle, ubiquinol, reduced form of ubiquinol, that is ubiquinol, first binds to the ubiquinone binding site here, and here one electron is transferred to the iron sulfur cluster, and as a result, it becomes a semiquinone. Semiquinone again further transfers the second electron to the cytochrome Bl. As a result, it becomes oxidized ubiquinone, and ubiquinone, and this ubiquinone again diffuses into the matrix side. Again, it binds to the ubiquinone binding site. Again, the electron that is coming from the cytochrome Bl, which in turn is transferred to the cytochrome BH, and the cytochrome BH in turn transfers this electron to the ubiquinone. As a result, it becomes a semiquinone. So that means the one ubiquinol finally it becomes semiquinone, but the two protons are transported here, and of course the electron that is transported from the iron sulfur cluster is further in turn transported to the cytochrome C1, that is the cofactor in the complex 3, which in turn transfers the electrons to the mobile electron carrier, that is the cytochrome C. So that is the first half of the Q cycle. So coming to the second half of the Q cycle, in the second half, again you can see here that in the second half of the Q cycle, what happens is in the second half, again another molecule of reduced ubiquinone that is binds to the QP site. Again, one electron is transferred to the iron sulfur and becomes the semiquinone, and two protons are transported to the intramembrane space, and the UB semiquinone further transfer another electron to the cytochrome Bl, and again it becomes the ubiquinone, and this ubiquinone again enters into the ubiquinone pool, and here the electron that is transferred to the cytochrome Bl again, which in turn further transfers to the cytochrome BH. The cytochrome BH again transfers this electron to the semiquinone that is formed in the first half cycle. As a result, the semiquinone becomes again ubiquinol. So by taking the 2 protons from the matrix, so that means here what is happening here is that so two reduced form of the ubiquinone molecules are utilized here. That means 2 ubiquinol molecules are utilized here, but the 1 ubiquinol is regenerated. Only 1 ubiquinone is utilized, but the 4 protons are translocated across the membrane, and the cytochrome 2 cytochrome C molecules are reduced. And you can also see here what exactly happens here is that when the ubiquinol is oxidized to ubiquinone, and as the electron is transferred through series of iron sulfur clusters and the cytochromes, and finally the cytochrome C is oxidized cytochrome C is converted into the reduced cytochrome C, which is a mobile electron carrier. So this process takes place in the form of 2 half cycles. This is known as the Q cycle, and this is about this one. Coming to the complex 4. Complex 4 is also known as the ATP cytochrome C oxidase, and this complex 4 so this complex 4 is also involved in the proton translocation, as I already said in the previously. And here what the complex 4 has many subunits, and see it has the 3 redox centers. So it has many subunits, that is subunit 1, 2 and 3, and there are 3 redox centers in the subunit 1, and it has the 2 hemes and the 1 copper atom. And you can also see here the copper atom is close to the iron atom, that is heme A3, that forms a binuclear center. So when you look at this diagram, you will understand that this is cytochrome C oxidase, that is the complex 4, and it has more than 10 subunits, and it has a different prosthetic groups like heme A, heme A3, copper A and copper B proteins. And it has, as I already said, that it has this enzyme has 3 subunits. You can see here that this is subunit 1, 2. This is the subunit 1, and this is subunit 2, and this is the subunit 1 is embedded in the membrane. You can see here this is the membrane, and it has the 12 transmembrane alpha helices and the 3 redox centers. As I already said that 2 redox centers are heme A and heme A3. This is the heme A, and this is the heme A3, and the third is the copper B. You can also see here this is the copper B, and the subunit 2 has 2 transmembrane helices. It forms a beta barrel domain on the exterior surface, and it has only the copper A site. This domain has only copper A site, whereas the subunit 3 has 7 transmembrane helices, and it has no redox centers. And if you see here that what exactly happens here is that the reduced cytochrome C, that is a mobile electron carrier, that this reduced cytochrome C binds to the subunit 2 and transfer those electrons to the copper A site at the beginning, and this copper A site accepts 1 electron at a time. As I already said that the copper ubiquinone, all these copper, all these can only accept 1 electron at a time, and therefore for complete reduction of oxygen to the water, we need 4 electrons. That means 1 NADH can only release 2 electrons, but that means the 2 electrons can only reduce half O2 plus 2 electrons plus 2 H plus becomes H2O. But complete reduction of 1 O2 requires 4 electrons. So now here, so when the electron, that means cytochrome C initially passes electrons to the copper A site. From the copper A site, this copper A site it moves to the these copper acid, it moves to the copper the cytochrome A. From cytochrome A it goes to the copper B, and copper B which is close to the cytochrome A3, and this is the binuclear complex, and near the binuclear complex it is the site for the reduction. That's where the oxygen is reduced to water, and so that's what takes place in the complex 4, and therefore the net reaction that takes place in the complex 4 is that the cytochrome C, reduced cytochrome C, undergoes oxidation, and whereas the oxygen undergoes reduction to water. As I already said, that one NADH can only can transfer only 2 electrons. So it can cause the but in order to complete reduction of one molecule of oxygen, we need four electrons and the 4 protons. That means tetragon reduction should takes place. So therefore, when one NADH, when it releases 2 electrons, that means 2 cytochrome molecules. The cytochromes are also the 1 electron carriers. So the 2 cytochrome molecules, when the cytochromes are releasing 1 electron at a time, so it can transport 2 protons for the pair of electrons that are transported to the molecular oxygen. And so this is about then last complex, that is the ATP synthesis, and this ATP synthase is a complex of 5, and this ATP synthase is also known as the complex 5, and that's where the ATP synthesis takes place. And this ATP synthase catalyzes the synthesis of ATP from ADP and PI, and it uses the proton gradient generated during the electron transport chain, and it is a multi-enzyme. It has many subunits, and you can see here it has it has mainly two parts, F0 subunit and the F1 subunit. And the F1 subunit has the catalytic activity. That's where the ADP is converted into the ATP. And you can also see here the F1 subunit has the F1 complex has many subunits, and the F0 complex has again many subunits. And the enzyme is, you can see here, the enzyme is embedded in the inner mitochondrial membrane, and you can also clearly see here that it has F1 subunit, and this is the F1 subunit, and this is the F0 subunit, and the F1 subunit has a knob-like structure, whereas the F0 has a stag. And so you can also see here that the F1 subunit has 3 alpha, the F1 complex has 3 alpha subunit and 3 beta subunits, 1 dou subunits, gamma subunit, 1 epsilon, whereas the F0 has so 1 A1 subunit and 2 B2 subunit, and 10 to 14 C subunits are present in the F0 complex. So that means it has F1 complex and the F0 complex. F1 complex is a catalytic part, and the F0 complex is embedded in the membrane. And if you see carefully here that this enzyme, all these complexes, mainly these, you can see here, the C subunit, and the gamma subunit and the epsilon subunit, C, gamma and epsilon subunit, they form like a rota, and whereas the A, B, you can see here, this is A subunit, this is B subunit, and alpha 3 and beta 3 and dou, they form like a stator. So now, so what exactly happens here is that when the protons, as I already said, that in the electron transport chain, once again, you just see here in the electron transport chain, as the electrons are moving from NADH2 to the molecular oxygen through several oxidation reduction centers, the protons are transported from the matrix to the intermembrane space. As a result, the proton gradient is established, and this proton gradient is essential for the synthesis of ATP, because when these protons are again translocated back to the matrix by ATP synthase, the ADP would be converted to ATP. So as I already said that this ATP synthase has 2 parts, one is F0 and F1 is the F1 component, and so as I already said that it has it has many subunits. So and it has a stator and the rotor. So what happens is that when the protons are moving from the matrix, from the intermembrane space to the matrix, and the rotor rotates, and that brings a conformational change in the stator, especially in the beta subunit, and the beta subunit has the ATP is activity that converts ADP into the ATP. You can also see here as the protons are moving. So the rotor rotates, and that brings a conformational change in this one, the beta subunit of the stator, and as a result, the ADP is converted into the ATP. So that's what this takes place. There are that's what takes place in the case of this ATP synthase, and we know that the proton translocation, when the proton is translocated from from the intermembrane space to the matrix, the ADP is converted into the ATP, and so that means the synthesis of ATP is coupled to the translocation of protons. So that's why this process is also known as the oxidative phosphorylation, because in this process the ATP is synthesized when the electrons are transferred from NADH and FADH to the molecular oxygen through several oxidation reduction centers. As a result, the proton gradient is formed, and this proton gradient is utilized for the synthesis of drives the ATP synthase. And this is also known as the chemiosmotic theory proposed by Peter Michel. So, according to chemiosmotic theory, during the electron transport chain, proton gradient is generated, and this proton gradient is essential for to drive the ATP synthesis by the ATP synthase. So once the ATP is synthesized in the in the mitochondria through the process of electron transport chain, this ATP has to transported into the cytosol, because the many reactions takes place in the cytosol. So therefore, in order to transport this ATP into the cytosol, again this uses the cell has another mechanism by having the ATP ADP translocase. As the ATP is transported out of the matrix into the cytosol, ADP enters. So during this process, you can see here ATP has 4 negative charges, whereas ADP has only 3 negative charges. That means ADP has 4 minus charge and ADP has 3 minus charge. So there is a charge difference. And in order to compensate this, the one more proton also enters into from the intermembrane space to the matrix by another that is the phosphate proton symporter. And of course, the formation of the ATP from ADP and PA also requires another from ADP. Formation of ATP also requires ADP and also another PA. That's why, so when the ATP leaves the cell from the matrix to the cytoplasm, and there is a charge difference, and there is a net -1 charge difference, and in order to compensate this, one inorganic phosphate and the one proton enters into the matrix. So this is about the ATP ADP synthase. And now last one is the P by O ratio. P by O ratio, it is nothing but the molecules of ATP formed in oxidative phosphorylation per the electrons are when the electrons are transferred from the NADH2 to the molecular oxygen. And the P by O ratio is, we know that when NADH is donating its electrons to the molecular oxygen, 10 protons are pumped out. So 10 protons are pumped out, and in order to synthesize one ATP molecule requires 3 protons should enter into the matrix through ATP synthase. but one proton is also required to transport the ATP out of the cell. So therefore, 4 protons are utilized for the synthesis of one ATP molecule, as well as the synthesis as well as the transport. So therefore, the P by O ratio for the NADH is 10 by 4, that is equal to 2.5. So 2.5, that means for there are 10 protons and 4 protons are required for the ATP synthesis as well as the ATP exit from the matrix. Therefore, the P by O ratio is 2.5. So uh, so this is the so in case of FAD, you can see here in case of FAD and the FAD, you can see here the FAD is a complex 2. So in the sense, FAD enters into the electron transport chain at the complex 2. So therefore, it has only only 6 protons. Only the 6 protons are only transported when FAD donates its ion electrons to the molecular oxygen. Therefore, the P by O ratio for the FADH2 is 1.5. So this is about the P by O ratio. In tomorrow's class, we will discuss the remaining part of this one. So that is the inhibitors of the electron transport chain as well as the shuttle mechanisms.